Helicase or helicase are enzymes that unwind the parental double helix by separating the two strands of nucleotides. Helicase break the hydrogen bonds that link the complementary bases which leave the leading and the lagging strands to begin the DNA replication process. After helicase splits the DNA strands, single-strand binding proteins, otherwise known as SSBPs, stabilize the separated parental DNA strands and keep them from re-pairing. SSBPs are enzymes and proteins that attach to the individual strands to hold them apart and prevent twisting.
Primase is an enzyme that synthesizes short RNA primers using the DNA as a template. These primers serve as a starting point for DNA synthesis. DNA polymerase III adds nucleotides to the primer on the lagging strand, forming the Okazaki fragments. The main function of the third polymerase is duplication of the chromosomal DNA, while other DNA polymerases are involved mostly in DNA repair and DNA synthesis.
DNA polymerase I is what removes the primers on both the leading and lagging strands and replaces the primers with DNA nucleotides. So once the primase comes through and lays down all the primer, this is what removes that primer and replaces it with DNA nucleotides according to base pairing rules. DNA ligase is the enzyme that joins the Okazaki fragments together on the lagging strand by joining the sugar phosphate backbones to create one continuous DNA strand. So once these fragments have been created, this is what goes over all of them and connects them and to make one smooth continuous DNA strand.
This is topoisomerase, and what this does is as the helicase expands on both sides, the parent DNA gets tighter and tighter. So this cuts the parent DNA, so it doesn't get so tight where the helicases can't expand anymore.